So let's see how water isotope analysis is actually made. And it all happens inside this very boring looking black box. And inside that box is a laser system. And the isotope analyses work because the different masses of oxygen and hydrogen absorb laser light at slightly different wavelengths. So if you design an instrument where you have the right laser and the right laser wavelength to interact with oxygen and hydrogen, it can then count how many oxygen 16 and oxygen 18 there are, and how many hydrogen 1 and hydrogen 2 there are, and work out the isotope ratio. So let's see analysis in action. So the water samples are in these little blue vials, and we're going to pick up a sample, we're going to rinse the syringe a couple of times, and then inject the water into the syringe, and then the syringe is going to move over to the sampling area, where it's going to vaporize that water into vapor so it can be injected into the machine. So the water sample is now inside that blue box with a heat symbol on it, and it's being warmed up and vaporized. And once it's turned to vapor, it's going to be drawn very slowly through the plastic tube into the back of the machine. You can hear the pump that's sucking through in the background. So now our sample is now heading off into the machine, into a large cavity, a chamber inside the machine, where the laser is now going to bounce around in that chamber, interacting with all the water molecules to work out its isotopic composition. So we can see what it's doing now. It's measuring the spectra. Here's some data that we've had from before. And this is what the machine's actually doing. You can see it's got some absorption peaks there. It's trying to line up the absorption to the different isotopes, and then it's going to count them all. It takes about 90 seconds to make an analysis, and then the water sampler will start again, and we'll go and inject the next sample. And we tend to make about 5 or 6 analyses on every single vial, so it's going to measure the same one again. And then take the average, you get a reproducible result. So here we go again, next sample. And here's the results of the last sample just coming in. So we have some flags there which says, is there anything going wrong? And they're green, so it's good, it's normal. And we have a column of data that says how much water was measured, the number of molecules, and then the isotopic composition.